also been provided. I want to thank you for joining us, and now I welcome the Dean of UCI Law, L. Richardson.
Good afternoon, everyone. It's my pleasure to welcome you to this special presentation in UCI Law's COVID-19 and the Law series. And this afternoon, we'll be discussing the state of current healthcare innovations to combat COVID-19. And we are so very fortunate to have Dr. Hans Keirstead and Senator Joe Dunn to share important insights on the battle against this virus. Before I introduce Dr. Keirstead and Senator Dunn, I want to recognize the team from both UCI Law and from AIVITA Biomedical who helped make this event possible. And they are Matthew Bayless, Rebecca Bergeron, Jillian Henry, Dennis Sloan, Michelle Shannon, and Marianne Soden. Thank all of you for pulling together this remarkable program. It couldn't have happened without you. So let me turn now to our fabulous speaker, Dr. Hans Keirstead. Dr. Keirstead is an internationally known stem cell expert, and he has led therapy development for late-stage cancers, immune disorders, motor neuron diseases, spinal cord injury, and retinal diseases. His work is truly groundbreaking and remarkable. He is currently the chairman and CEO of AIVITA Biomedical, and he has founded 4 successful biotechnology companies, each returning significant value to investors and, importantly, treatments to patients. He also sits on the board of several prominent biotechnology companies. Dr. Keirstead is also one of our own. He was a full professor at UCI, where he founded, directed, and then erected a building for the UCI Stem Cell Research Center. He's also a founding advisor of the California Stem Cell Initiative that resulted in a $3 billion stem cell fund. He's also been a longtime advisor to several governments on biomedical policy. Dr. Keirstead received his PhD in neuroscience from the University of British Columbia, where he received the Cameron Award for the most outstanding PhD thesis in the country. Additionally, Hans received the distinct honor of being elected as a Senate member of the University of Cambridge and also as a fellow of the governing body of Downing College. Unsurprisingly, he was the youngest member to have been elected to both of these positions. And personally, I'm so grateful to Hans because he agreed to serve on the law school's Board of Visitors, where he brings his perspectives as a brilliant researcher and a successful business leader to all of the initiatives that we have here at UCI Law. And in addition to that, it was through our collaboration with Hans that we began the very successful CEO Fellowship Program, in which our students now work closely with CEOs and general counsel in companies throughout Orange County, including at AIVITA Biomedical. So I want to thank you so much, Hans, for offering to share this important information with our law school community today, and additionally for always saying yes when I reach out to you.
My pleasure.
You are an indispensable member of our UCI Law family, and I'm so very grateful for your support. Thank you so much.
Thank you.
Now, today's presentation is moderated by Senator Joe Dunn, who is one of the founders of our law school. And Joe worked tirelessly for 20 years ago to bring the law school to UCI. And since we opened our doors 11 years ago, Joe Dunn, Senator Dunn, has been a vital part of our UCI Law community. Currently, Senator Dunn serves as special advisor to the dean and as a lecturer at the law school, teaching courses including Pushing the AI Frontier, its impact on law, policy, and the legal profession, as well as the very popular course titled, So You Want to Change the Law? Mastering the Legislative and Congressional Process and Politics. Prior to entering the Senate, Joe practiced law, and he handled products liability litigation involving defective medical devices and pharmaceuticals, as well as environmental cases. He worked in the Newport Beach office of Robbins Kaplan after law school, And afterwards, he joined Robinson, Calcagney, and Robinson, where he continued to focus on products liability and complex litigation until he was elected to the Senate. Senator Dunn was a member of the California State Senate from 1998 until 2006. And while he served on the Senate, he chaired key committees, including the Judiciary Committee and the Housing and Transportation Committee. Between 2006 and 2014, Senator Dunn served as CEO of both the State Bar of California and of the California Medical Association. He received his JD cum laude from the University of Minnesota Law School, and it's only because UCI Law didn't exist yet that he went and his BA magna cum laude from the College of St. Thomas. So Hans and Joe, Dr. Keirstead and Senator Dunn, thank you so much for taking the time to be here this afternoon. I know I look forward to the conversation. I know all of our participants do, so I'm going to end now. And with all of the people who have joined us this afternoon, please join me in giving a very warm welcome to Dr. Keirstead and Senator Dunn.
Thank you, Song, very, very much. Hans, I'll only take a second here, and then we're going to turn over the microphone and spotlight to you because you're the main event, of course, here today. I have to share one thing, Song, that I shared with Hans before, when both his Canadian roots and my Minnesota roots make us uncomfortable with introductions like that. And there's an old saying that Harry Truman, when getting a nice introduction, would always say, "An introduction my father would appreciate and only my mother would believe." So anyways, Hans, a gigantic thank you to you. As Song had just indicated, on behalf of all of us at the law school, you've been such a dear friend for me personally for a very, very long time, but to the law school almost from the beginning as well too, Hans. But I also want to welcome our audience. We have a very diverse audience of professions and so forth, which we welcome you. And Hans, at this point, they're all here to hear from you. So let's turn the microphone and spotlight over to you, everyone. Please welcome Dr. Hans Keirstead.
Thank you very much, Song, and thank you, Joe. I'm just delighted to be here and, play a role in this fantastic institution. I've been a fan since its inception. So I'll share a screen here and run through a few slides that I think speak to the real cutting edge of what is happening right now in the world, and of course, these views do not reflect the views of the University of California at Irvine or President Trump administration or anything else like that. They're mine, and I've pulled from a number of sources to get them, and I'm delighted to give you this perspective. So, very appreciative here to share updates on COVID-19 treatments and vaccines in particular. With such worldwide need and such attention, and an unprecedented amount of third-party monetary support, we're seeing just an unbelievable amount of activity from the scientific and medical sectors right now. There's never been such a coalescence since putting someone on the moon, frankly. What would normally take decades of effort is being condensed into months. And as you would expect in such a situation, there's a lot of noise and a lot of nonsense out there. So in this talk, I've done my best to separate the wheat from the chaff, so to speak. So over the past few months, we've seen what I feel are three major sources of influence coalesce to promote containment. Quite clearly, the biology of one single virus has dominated the various influencers. And it did so to a degree that humanity has just simply never seen before. Biology said, distance yourself from the others to reduce spread of this virus. And culture said, I fear for the life of my partner, my child, my family, myself. And the economy said, all commerce involving personal interaction must cease to protect the taxpayer. But that has dramatically changed. Biology cries out the very same message that it did last month, containment. But the economy, it literally cries. Indeed, we've seen much of it die. We simply cannot sustain this nation's well-being by continuing our stay-at-home policies. And human nature, human culture has drifted away from the influence of biology as well. We're social creatures and we grow tired of containment. So it's rational that economic drivers prevail at this time. But we must develop, and until we develop a treatment or vaccine, we will have to accept the herd mentality and the culling of human and life that it entails. So this bug is not going away, neither in the outside world or in the United States. COVID-19 is likely to consistently thin our population until a treatment or a vaccine is made. While the scientific and medical response has been nothing short of heroic, the fact remains that our scientific and medical community is working from a relative point of ignorance. And that lack of data is going to result in a lot of false starts and fast failures. Spread, as you can see from this graph, updated to May 9th, is still increasing. Reopening is showing resurgence. Just last week, Tuesday to Tuesday in Orange County, we've got record deaths increasing. We saw the highest number of hospitalizations and ICU patients and the highest number of new cases. In every single jurisdiction that has lifted restrictions and is starting to open up again, we are seeing a resurgence of this virus and the associated death and disease. And a recent study helps to explain this. Speaking causes the virus to remain airborne for up to 14 minutes. Just holding a conversation, 14 minutes, that virus can stay airborne. And masks are certainly effective to prevent that. A month ago, there were about 300 planned or active clinical trials. Now there are over 1,500, from 300 to 1,500 in 1 month. But let's be clear, the availability of diagnostics for disease or recovery are not readily available. There are no completed clinical studies showing that any potential treatment improves outcomes in patients with either suspected or confirmed COVID-19. And there are no clinical trial data supporting any vaccine to prevent you from getting the disease. But in a remarkably short period of time, we have some developments in each of these categories. And although this slide shows only a few dozen treatments or trials in active testing, there are over 1,500 in the category of planned or active. So let's jump into each category. Almost every test in development to determine whether you have COVID-19 looks for bits and pieces of the virus in your respiratory pathway. These tests are coming along well, with many authorized by the FDA, as you can see. But these are currently only available for healthcare professionals. Importantly, about 50% of infected people do not show symptoms. Readily available tests would help us identify and isolate carriers. And if this virus can't find a new host, it dies. Isolation is an extremely effective method to eradicate viruses. We need to know who to isolate. We also need to know who already has had the disease by assessing antibodies to the virus in the bloodstream. Viral infection usually results in the development of antibodies in the bloodstream, which then confer resistance to further infection. It is believed that antibodies to other coronaviruses like SARS and MERS last from 1 to 3 years. Clearly, resistant people are not likely to get the disease while they're resistant, and they're not likely to be able to spread the disease. That's a heck of a get out of jail card. Although the number of available tests is slowly increasing, almost all of them are still limited in their availability to healthcare professionals. So it's important to note that these tests have limited efficacy as well. Not only availability, but how well they work and what they can tell you. Although they tell us if you have antibodies to the virus, they do not tell us how much you have. How many antibodies are enough? Are you just building your immunity during an active viral infection? And with this particular virus, we do not know if antibodies do provide resistance or for how long. And since we don't know that, we also don't know if plasma from one person containing antibodies can protect another if plasma is transferred. But clearly, these tests are an incredibly important tool for our economic recovery, as they will help us to determine when an employee can get back to work and how long they can stay there, knowing that they can't get infected and they can't infect others. So there's a desperate need for these tests to become widely available, both tests to detect whether you have the current disease and tests to determine if you have previously been infected and now have resistance. Had this disease been acknowledged, these tests maybe would have been more widely supportive. So had they been more widely supportive and distributed by our government at the onset of this pandemic, the scientific and medical community would know infinitely more than we do now about disease transmission, allowing for a much more refined management of viral spread and personal protection, but also a more refined management of economic shutdown and the return. Fortunately, the private sector has made some advances and seriously stepped up with regards to treatment of COVID-19. These advances are moderate, but they do underscore the tenacity of our scientific and medical industries in this nation. It's really been quite remarkable. Clinical testing suggests that transfer of plasma from a previously exposed person to a COVID-19 victim may confer benefit. And this treatment of plasma transfer is approved for use in emergency situations, such as healthcare professionals with disease. It's important to note that this is not an ideal treatment to prevent disease, as you would have to be taking antibodies constantly. And of course, they're little proteins that degrade in your blood system. You know, earlier this week, I listened to the CEO of a San Diego-based antibody company shamefully promoting that his company can treat the world and that a cure is now widely available. Why are you waiting? Come buy my antibodies. Not so. Indeed, a recently published paper indicated that about 30% of recovered patients show low antibody titers, with many showing antibody levels that are below the detection capability of the assays. We really don't know whether or not they confer and how long they confer, and having to take antibodies constantly to prevent disease is just not a tenable situation. Also notable is the issuance by the FDA of emergency use authorization for remdesivir for the treatment of COVID-19. In the United States, remdesivir is an investigational drug that has not been approved by the FDA, and the efficacy of remdesivir for the treatment of COVID-19 has not been established. The distribution of remdesivir in the United States has been authorized, but only for the treatment of hospitalized patients with very severe COVID-19. And interestingly, the Japanese Ministry of Health, Labor and Welfare recently granted regulatory approval of remdesivir as a treatment for COVID-19 under an exceptional approval pathway that they have in that country. The studies of remdesivir indicate a shortening of hospital stay by about a third, and only unfortunately a suggestion of efficacy in decreasing mortality by about 25%. But that suggestion of decreased mortality did not reach the point of statistical significance at all. So you would think that SARS and MERS treatments would be potential candidates for COVID-19 treatment. But although we saw a great success in a dish, as we're seeing now for SARS-CoV-2, none proved to benefit humans. Likewise, the HIV and malarial drugs that have not shown any benefit and are known to cause some very serious safety concerns, such as the induction of heart problems, they have also failed in testing. Highly accredited laboratories in several countries have proven that malarial drugs do not work for COVID-19. This suggestion that they did, and the laboratories and doctors that did it were in France. The entire country of France has banned the use of this drug for the treatment because it has been definitively shown not to work. But probably far more important than treating the disease is preventing it in the first place. And that is a very, very difficult task. Viruses are so good at evading our immune systems and infecting us that about 5 to 8% of our genome is actually viral. The best weapons that humanity has ever developed against these invaders are vaccines. Vaccines typically mimic a small bit of the actual virus, training our immune systems to recognize it, allowing for the development of our immunological armies before we ever experience the real beast. And they work. There's absolutely no doubt that vaccines work. It's just that they're extremely difficult to make. Just think about how few vaccines are available right now and how many bugs there are in the world killing people. Over 90% of vaccines in development fail. That's 90% in the clinic. About 96% of vaccines preclinical fail to get into the clinic at all. So 96% failure on the way in, 90% failure while you're there. So we know who the enemy is, but the status of our weapons development is just barely beyond the point of sketching weapons blueprints on a piece of paper. The vaccines that are currently in development for COVID-19 are largely of the typical type: attenuated or incompetent virus, bits of virus, or their genetic material. I'm honestly very worried about these approaches. With regards to the attenuated viruses, incompetent virus or bits of virus, we run into the problem that the SARS-CoV-2 virus has a trick of coating itself in sugars, creating this invisibility cloak to the immune system. Vaccines comprised of incompetent SARS-CoV-2 virus or bits of virus are very likely to similarly get coated because they are coronaviruses that do that. Viruses comprised of incompetent SARS and MERS did the same thing. They coated themselves. Coated viruses can actually suppress the immune system by these little siglet receptors on cells, antigen-presenting cells of the immune system, creating tolerance of the infection rather than cytotoxicity. So these types of vaccines run the risk of inducing tolerance and downregulation before a real infection occurs. The genetic approaches, on the other hand, get around this problem by inserting genetic material of the SARS-CoV-2 virus directly into our immune systems. But these types of viruses have a challenge of delivery. How do we effectively deliver genetic material to our immune cells, and only our immune cells, without creating toxicity in every other cell type that sucks them up? And if our immune cells are correctly targeted and taught to detect that little protein product of the inserted gene, usually a spike protein, spike 2 of the SARS-CoV-2 virus, that's only one target. One target that may be hard to find, and one target that may mutate, as has been shown to mutate in the very recent past in this very virus. Speaking to these concerns, it's important to note that there are no such vaccines currently approved for human use in any disease, and all of them failed in SARS and MERS. Moderna, Pfizer, and Inovio are 3 large companies currently testing such vaccines that insert genetic material. These types of vaccines have huge benefits for scaling of manufacturing, but are faced with the several challenges like the ones I just mentioned. The Moderna RNA vaccine was shown to produce antibodies in their phase 1 trial just last week that ended, although at levels that approximate antibody levels after natural infection. Vaccines usually produce antibody levels that are much, much higher than antibody levels after natural infection. And the likely toxicity results in the necessity of 2 dosings a month apart. It's likely because they have to lower the dose to lower toxicity, and therefore they're getting a lower effective dose, so they need to do a booster a little bit later. The University of Oxford, in collaboration with AstraZeneca and distinctly Johnson & Johnson, are 2 groups that represent another type of vaccine. It's a harmless carrier, an adenovirus or a lentivirus that's been genetically engineered so it can't replicate, but it does carry, through genetic engineering, the SARS-CoV-2 spike 2 protein. This type of vaccine has had one single success in all of our history in generating an effective vaccine for Ebola. In J&J's case, this represents a $1 billion initiative between Johnson & Johnson and BARDA, the government's BARDA, to scale up production even before efficacy of the potential vaccine is demonstrated. Last week, Oxford released the results of their monkey preclinical trial, revealing that their vaccine failed to induce protection in monkeys. Every vaccinated monkey caught the disease, just as the unvaccinated monkey did. Although in the vaccinated monkeys, the severity of the disease was decreased a little. And it's just extraordinary that they're not making modifications and pressing ahead into human clinical trials with a vaccine that does not cause protection, but it does cause perhaps a slight lessening of disease. And honestly, we wish them all luck. It's important to note that there are no competitors in this race. No single company can generate the manufacturing capacity to treat the world's 8 billion people. A couple of these companies have boldly announced initiatives to scale manufacturing to treat 1 billion people over several years, if they put in several $billions of manufacturing scale up. Once we find effective vaccines, production will be limited by the high cost of technology to scale up. Government demands placed on companies to preferentially treat peoples of their own nation, and the incomprehensible economic drive to preferentially treat first world nations. And honestly, I really fear that the vast majority of people on this earth of limited means and in countries with limited capabilities will go without vaccines. And let us hope that those few multi-billionaire angels walking amongst us step in and inspire us all. So as you navigate the next few months, while the scientific and medical communities work to create vaccines and treatments, please do not take your eye off the biology of this disease. Economic drivers certainly prevail at this time, and cultural issues will certainly conspire with the economic drivers to push people out of their protective homes. Please keep your eye on the biology, which incessantly speaks of recurrence, mutation, and further spread. The potential for success in the short term compared to previous pandemics is dramatically elevated by nearly a couple hundred years of concerted virologic research combined with exponential advances in tech. As we collaborate globally in real time, sharing findings, we are drawing upon multiple approaches and building an arsenal of vaccines. Every type that can be made is being made. And in a world where 90% of vaccines fail, we need multiple approaches to protect us from this virulent contagion. We cannot limit ourselves to thinking of vaccine development as a competition with one winner. We must approach this with a robust understanding of all mechanisms of protection and consider all technologies. To win this battle against SARS-CoV-2, we're going to need an arsenal. Not one vaccine, not two or not three winners, but every single comer. And all over the world, that arsenal is being built right now. So thank you very much. Appreciate your attention.
Sorry, Hans, I had to come off mute and get back on video. Great presentation, Hans.
Thank you.
I want to get right to the questions that we're getting from our audience. And you can imagine if I get through all of these before our time is up, I've got about 300 questions of my own. But we'll reserve my cross-examination of you, Hans, until later.
Please.
So I want to go right to our audience questions that have been forwarded to me by both Matt from your team and Marianne from the law school, of course. I'll paraphrase them a little bit, but let's assume a vaccine is rushed through to approval stage. Let's just make that assumption. From your perspective, how long would it take to determine if there were unexpected side effects and even note that there were side effects, yet let alone what to do about those? Comments?
The good news here is that safety with a vaccine is detectable within weeks. We're not talking months or years. So it's very, very easy to take a blood draw and see if antibodies have been made. A vaccine will often produce antibodies in your blood within 3 days, and the average is 7. So very, very quickly, we're going to know if this thing is safe. And then we're going to have to watch out and be careful of these expedited approvals, because a whole bunch of people might be taking a whole bunch of vaccine that doesn't have any efficacy, because efficacy requires a lot more work and a lot more time. Clearly, the best test of efficacy of a vaccine is to be exposed to the SARS-CoV-2 virus. In this environment of a world's pandemic, with Operation Warp Speed going and the just dramatic need out there, it is unlikely that the FDAs of the world, the regulatory bodies, are going to insist upon very lengthy challenge assays where they subject a person in clinical trials to the disease. Rather, this is going to come from post hoc, where someone lives in like a city in Brazil right now in densely populated areas with skyrocketing levels of SARS-CoV-2 infection, and they have antibodies going into it, and they come out of it without disease. It's going to be real world experience. So that's going to be slower, but the good news is the safety's fast.
Got it. Thank you, Hans. Let me go on to another audience question that it actually touches upon an issue, Hans, that I think all medical scientific researchers in the world face many times. Now we're dealing with it in a pandemic situation. But the question is as follows. Given that there are many tests that are in development that you've commented on in your presentation, why not try to distinguish those and put more of our research effort into those that can generate results faster, even if the quality might not be as good? That balancing act. Your comments on that?
Well, you know, that's actually being done. I think every single potential vaccine is being supported and rushed forward right now. There isn't a sorting out, because frankly, there's not enough data. They're all in very, very early stages, so there is no mechanism to sort them out. Every one of them does, thankfully, have to go through regulatory bodies, and those regulatory bodies, like the FDA, are going to shut them down if all they have shown is safety and there is no efficacy and they're just taking everybody's money. If there is any fear of safety, they will not be, like ill effects, they will not be advanced. So I think actually the commercial world right now is sorting this out. All avenues are being explored right now, and that's exactly what we need in the face of this pandemic, so that's good. Now we're moving into a time when the bogus ones need to be shut down quickly. And because everyone's eyes are on these things, and there's some pretty big players, and the regulatory agencies are all involved, I think we will see relatively quick shutdowns of the bogus efforts.
I'm going to do moderator's prerogative here. I want to do a follow-up on that question. As you know well, and you've shared with me over the years, as the stem cell science has developed and moved into treatments and so forth, you've unfortunately seen many moves to push alleged stem cell-based treatments out both in the US and around the world, and you've seen a lot of those, unfortunately. My follow-up question is, how long does it take a regulatory body to deal with folks who are out there selling a treatment based upon, in this case, it would be treating COVID virus, but how long does that regulatory process take to catch up to those folks?
You know, Joe, great question, and the answer is just saddening and maddening. You know, you mentioned stem cells and vaccines. I just yesterday became aware of a group, I believe it's in Seattle, that's saying we're curing COVID-19 with a stem cell injection. Like, you know, my field of stem cells, honestly, 19 out of 20 of the things you hear about are completely snake oil. And that's certainly one of them. Case in point here, over 300 stem cell purveyors in the United States were caught by the FDA. They were bogus. They were all sent cease and desist letters by the FDA. But then nothing happened. The FDA can look at you, they can shake a stern finger, they can write you a letter, they can post your name on a site, you're bad. Everybody should look on our site and see that these guys are bad, but they don't have a police force behind them. They don't have any ability to then march into that town in central United States and shut that clinic down. And that clinic continues to suck out $40,000 per naive individual that walks in their door. It's a really horrible thing. So we are going to see this, unfortunately, where we're going to have fake vaccines hitting the marketplace, and good people are going to be paying a lot of money for a bad vaccine, and the FDA may come down and send them letters, but the ability to then enforce and stop those people is very, very limited.
It's unfortunate, obviously, particularly when we're in this pandemic environment. I want to pose a question from another one of our audience members, someone you know well and I know well, a dear friend of both of ours, in another space that we work together at that I won't mention, but and he's really posing it on behalf of the average person watching all the debate about what is COVID and so forth. And so it's a twofold question, but they're related. What is the difference between COVID-19 virus and a common winter cold? And then the follow-up, why does my annual flu shot not cover COVID-19?
Mm-hmm. Well, the first one, the difference, they're both coronaviruses. So the common cold, SARS, MERS, and SARS-CoV-2, they're all coronaviruses. But this little beastie, SARS-CoV-2 that makes COVID-19, had a mutation in its anchor. What happens is this little ball has a bunch of little needles, and they're like a barbed hook. Think of it as like a burr, and the virus hits a cell surface and it sticks its hook in, and then once it hooks, there's another little protein that punches through the membrane and inserts the DNA. So it's this little 1-2 punch, hook and then insert. And that little connector, the hook and insert, was what mutated in this virus. So it can grab hold so much more virulently and stay there and cause the infection and destruction of that cell, propagate to the cells surrounding it that destroys them, and then they propagate more and on, much, much worse than the cold virus. So it was just a simple mutation, frankly. And the fear here was that this coronavirus would mutate as fast as the cold virus. And if that was happening, ladies and gentlemen, this world would be in dire, dire trouble, because no vaccine could work. There's a reason why we don't have a vaccine against the cold virus. We can't make them fast enough, ramp them up and distribute them before those little beasties mutate, because the rate of mutation is so high. The good news about this SARS-CoV-2 is that the rate of mutation is actually moderate to low. So we will be able to hit it like we can hit a vaccine for the flu. So that second question, the flu virus is completely different in its genetic makeup, its shape, everything from coronaviruses. It mutates rather slowly, around once a year. And it is the production system for that particular virus type is so refined that it takes them a long time, but they can sequence the new mutated flu, insert that into the manufacturing system, make millions and hundreds of millions of doses, and ship them out to pharmacies all over the world where you can just go get a shot, all before it mutates. That's what we're trying to do with SARS-CoV-2.
And just to put a fine point on that, we got a question from one of our distinguished faculty at the law school, similar to what you were just commenting on. And this particular faculty member has family members who are on the front lines in the medical arena fighting the COVID crisis, and of course has connections around the world. And through all those connections, as we're seeing the news report, we're seeing relapses and new spikes after a decline. And the question really is, and here's the fine point on what you were saying, if we had a vaccine right now, how resistant is COVID-19 to it, and how quickly can the virus adjust and reemerge? Tell us a little bit about that timeline.
I think we're in good shape. If we get a vaccine that works, we will beat this thing. And then if you were to take that vaccine and I was to take that vaccine, we're no longer potential victims. So we cull the hosts out. there are fewer hosts. The more people that get vaccinated, the fewer hosts that can actually breed and propagate the virus. So that's why the flu goes away. There's a lot of people in the world that don't believe the vaccines work. They have some sort of conspiracy theory about them. They are incorrect. Vaccines do work. But it's those people, if there was enough of them not taking the vaccine, we could never get rid of the flu. It would constantly, 12 months a year, be infecting people all over the world. It's only because of vaccines that we're able to cull it back, because we rob that virus of hosts. So really good news here is that SARS-CoV-2 does not mutate very often. There's only a few variants now in the world over the last 4 or 5 months. So that's really, really good news, because if we did make a vaccine, and if the vaccine worked, now we will make vaccines. And the question will be, how long do they confer a resistance? They're going to increase antibody levels in one's blood. We're hoping that the antibody elevation is going to last for 1 to 3 years like it normally does. When you know how you have to get a flu vaccine every year? You're still carrying antibodies to last year's flu for 2 or 3 more years. And that one is getting killed out. But you are conferred a resistance for 1 to 3 years on average with a decent vaccine. If we get there with SARS-CoV-2, that little bugger is dead.
And another question from our audience that won't surprise you given what the comments you made. Obviously, you've talked about all the resources that are being put into the development of both diagnostic, treatment, and of course, prevention. But from Dr. Hans Keirstead's perspective, what do you think a best guess timeline is to the first effective vaccine?
This fourth quarter of this year, we're going to see a few, just a few, like meaning 3 or 4, maybe as much as 6 vaccines come off of phase 1 testing and be at a point in phase 2 testing where they've hit interim data. Interim data is a point where there's a possibility that you could detect statistical significance of safety and efficacy. So fourth quarter of this year, on the best case scenario, we may get to a point where not phase 3, but phase 2 testing. And for those of you who don't know, phase 1 is testing safety, phase 2 tests efficacy, and then phase 3 tests efficacy in a very broad number of people, looking for contraindications if somebody's popping heart pills and other drugs. Males, females, different races, etc. Phase 3 is a broader population. Phase 3 is always required. In this case, there is a chance, and I'm not saying it's for real, it's just that there is a possibility of expedited commercial approval. This is what Japan does. Japan's FDA, called the PMDA, has a couple avenues in it for expedited approvals, which they just did for remdesivir. They did not hold completed phase 2 or 3 clinical trials over there, but they just approved it. So there is the possibility that the United States FDA may analyze safety and efficacy out of phase 1 and phase 2, perhaps phase 2 interim only. And if those data look solid, may, and I'm recommending they do, expedite in a commercial approval for those for those vaccines, and then demand on post hoc analysis in the field. So you've got to still follow the patients. You still have to report safety and efficacy while you're commercializing. And if there's anybody harmed, if you show that efficacy isn't there, then you shut it down. But at least that way we get to the masses faster.
And Hans, as a follow-up to that question, it would seem from your very original slide of the 3 circles and of culture and economy and science, that until that vaccine is available, there has to be some level of protection for all of us, the masks, the social distancing. But again, as you well know, the public policy arena is trying to balance culture and economics, as you put on that. That would suggest to some degree or the other, we're going to have to be implementing safety measures within our communities until we got to that vaccine. Is that a fair statement?
It's fair, and it's absolutely correct. Like, honestly, you are foolish if you follow the economic drivers only and the cultural drivers only. You are foolish. There's only one thing that controls infection, and that is the biology of the disease. So you must keep your eye on that beast. Just because you're tired and culturally you can't stand it anymore, or just because the economy is opening up and you can go to the bar, to the restaurant and sit inside, follow the science. You will be propagating this thing. You will get infected. Infectious biologists predict that every human on this earth will be infected by SARS-CoV-2. It's just going to take a matter of time. We will all get it if it is not stopped with vaccines, and if it is not stopped by personal protection, keeping yourself closed in. And I can't underscore it enough. All of the data points to people over the age of 70 and people who have immune suppressed systems have a much, much higher chance of getting severe symptoms. And you're very much at risk. So please take this personal protection very seriously. I'm very convinced that we will see vaccine and effective treatments coming within 6 to 12 months, but that's a long time for you to stick to it and keep yourself safe and stop making your own body and those of your family a breeding ground to propagate this virus.
And Hans, we see a lot of news reports for us non-medical folks, and let's ignore the high risk factors that you just mentioned, over 70, for example. Can you explain to us in simple medical terms, why is it that some individuals who contract the virus are all of a sudden in intensive care, on ventilators, and others seemingly have minimal, if any, response? Why is that true?
The disappointing fact is we don't really know yet. No one knows. If you hear a doctor saying this is why, they're making it up. They honestly, the scientific and medical community has hints about why that is, but there is no evidence. There's one thing in you called IL-6, and there's some correlation between whether you have enough of it or too little or too much, where you may have susceptibility. There's a lot of correlation with vitamin D. I, for the first time in my life, am taking vitamin supplements, vitamin D only, because there's a correlation of people that have high vitamin D having no symptoms or very low symptoms. And then there's no, that's about it. The rest are very much not known. And a lot of it has to do with the genetics of the vaccine. Like, interestingly, in Europe, we were seeing children starting, that probably means a mutation, starting to come down with cardiovascular problems. And now we are seeing that in the United States. So we have probably followed the course of a mutation. So why older people, why younger people, why sometimes memory loss, taste, why the brain is affected, no one knows yet. It's very disappointing and it's very scary.
It is indeed. Another question came in from the audience just now. Of course, we're told over and over again to wash our hands, over and over, of course. Does that debilitate that hook and insert process that you described earlier as it relates to COVID-19?
The hook and process, no, but it blows that little bugger open. It's a ball of DNA with spikes sticking out of it. And the ball itself is disrupted. It's very, very weak proteins. And the smallest amount of detergent actually does blow it up and blow it apart. So it is very, very true that washing your hands and not touching this area of your face, putting the virus in close contact to your respiratory system, is what is going to protect you. And it's one of the most efficacious ways. The study I mentioned earlier about speaking, causing the virus to float in the air around you for 14 minutes. You and I are having a conversation 12 feet apart and we think we're okay. No, we're not, because air drifts and carries my exhaled breath over near you and you suck it in. If I had SARS-CoV-2 in there, now you have it. So masks don't protect you, masks protect other people. And it is very, very good social etiquette for you to be wearing a mask, because the symptoms only begin after you lose contagion. You are contagious before you have symptoms. This is one tricky little beast.
I want to ask, we're running out of time and we want to respect everybody at 1 o'clock, respect their time. I want to ask a difficult question. This is a moderator, it's prerogative, Hans. I want you to look backwards. We all know the old saying that hindsight's 20/20. And forget the public policy and political arena and all the debates that are there. From your perspective, looking backwards, is there something science field, medical field could have done in the number of years going back 5, 10 years to have anticipated this, or no, not necessarily? Just curious on your thoughts in looking backwards.
You know, the infectious disease world has been predicting pathogens constantly. There's not a biologist, a true biologist, which of course encompasses infectious disease experts, that doesn't feel that the most likely death and annihilation of the human species is a bug rather than a physical catastrophe like an asteroid. You know, there's a lot of evidence that human disruption of population in the past were all bugs. And we have, as I said, 5 to 8% of our genetics are viral. So we're living in a symbiotic way with viruses, and they mutate, and just like our bodies mutate, and mutations often result in death and propagation of that. I don't know if there's anything we could have done besides keep more funding for research. And so we have a basal level of research that is able to faster adapt, rather than having under-resourced laboratories and understaffed, rather than having a situation where youngsters looking at their professors say, oh man, that life is hard. Overworked and underpaid? No thanks. Rather than looking at them and saying, revered scientific and medical or professionals, I want to be that. And look what they can do. They can change the world. They can save humanity. All of that's true. It's just been a little neutered over the last couple of decades.
Unfortunately so. Hans, we've reached 1 o'clock. On behalf of all of us at the UCI Law School, we can't thank you enough. I know that Song is still listening in, and on her behalf as Dean and all of us, the family at the law school itself, we can't thank you enough. I'd like to thank our audience members for attending, and if this is the first time you've ever been introduced to Hans Keirstead, you know why we all love him dearly. One of these days, we're going to start the Hans Keirstead for President campaign, Hans. So on behalf of all of us, thank you, Hans. Thank you to our audience. Thank you to all the great professionals who made this event happen and pulled off without any technological glitches along the way. So again, thank you, everybody, and stay tuned for our next announced speaker series. And thank you again, Hans.
Go UCI Law.